innate, right? But here it's mentioned that the identification or recognition of the antigen is non-specific. So what does it mean by non-specific? No, it's not recording. Okay. Okay, in the meantime, before I forget, the attendance for today's meeting would be in the near future, guys. So please don't forget to take your attendance today.
Okay, Doktor.
Okay. So this is also recall your understanding about topic 1. So that's why I mentioned, although some of the topic in earlier part of this lecture will not be included in our final exam, but certain thing is just overlap, like definition of the innate immunity, right? The criteria of the innate immunity. You've learned in chapter 1, topic 1, but now we are going to touch a little bit here and there. So that's why probably some part that is related to this topic maybe come out in your final exam. Okay, so this is also the immunity type that is readily born with you. When we born, it comes together. You will not be improving over the time because it's not an adaptive kind of mechanism. So what most important in this subtopic you need to pay attention is that how do actually innate immunity does their recognition of the antigen. So before this, we've talked a lot about antibody bind to the antigen, detect the antigen, right? Which is basically the identification of protein toward a surface of an antigen. So in this topic, we are basically looking at the antigen, specifically which part of it, the molecular structure of it. So it's we know that the antigen, besides the foreign materials, may be also involve a unique molecule. Okay, before that, sorry. So, before a unique molecule that is transformed in our body tissues, the antigen that belongs to the microbials could become any part of an antigen. So the part that is could be recognized by the immune system, particularly in innate immune system, is called pathogen associated molecular patterns. So basically, under this type of immunity, they recognize the antigen specifically by the molecular patterns. So the term in short is PAMS. So if the immune system encounter any bacteria or any fungus, for example, they will know that they know that it is a foreign material based on a certain molecular pattern of the pathogen. So it's called PAMS. And we also know that certain antigen, apart from the foreign material, could be from our own tissues, right? So this is again, we go through in topic 4 on the antigen, hapten, and so on, right? So the PAMS could be also comprising of unique molecule displayed on stressed, injured, infected, and transformed human cells. Not only that, PAMS could be belongs to pathogens. It could be also belongs to our body tissue that has gone modification, alteration, like in a stress form. So they're not becoming normal, or they haven't they have not be they are not normal anymore, right? They become stressed, so they have changed their molecular pattern. So in a context of injured tissues, for example, or infected. So although infected tissue might be comprising of the pathogen, but the tissue itself could be also assumed by our immune response as a unique molecule or as a foreign molecule, known also as PAMS. And sometime in a textbook, you also come across a similar term known as MEMS, which is normally to refer the microbe associated molecular patterns to differentiate the pattern that is generally belongs to either the host tissue or the foreign materials. So both the pathogens and the host tissues that has gone alteration is referred as PAMS, a general term. But to differentiate them, normally, sometimes for the pathogen, we call them MEMS, micro-associated molecular patterns. So now that you have understand how innate immunity recognize what part of the pathogen they recognize pattern because they are unspecific. They use unspecific identification. Next is you need to know the example of the PAMS. So PAMS could be comprising of some structure or part of the pathogen itself. So it includes lipopolysaccharides. So these all of these criteria, I believe you have already familiar because normally you've learned this in the cell biology, in the basic biology subject in the earlier semester of this program usually. So we know that these are the criteria of the bacteria. They contain purines, lipoproteins, LPS. So these are example of PAMs. So our immune system recognize PAM because these are basically a protein that is not be available. That is not present in human tissue in the host tissue. Therefore, they know that this is foreign materials. So could be bacterioglycan, could be uh, lipoticoic acid if it's gram positive. So just now, if it was negative, gram positive, negative gram negative bacteria. So and the other part of the molecular structure of the bacteria, acid fast bacteria, could be lipoarabinomannan. So we can see, right, the pattern of the PAM is usually could be any part of the bacteria types, a unique part of the bacteria. So could be mannose, sequence, flagellin, protein of the flagellin, the bacterial and viral nucleic acid. So although we know that nucleic acid sequence could be shared by any species across different types, right? Human, animals, bacteria, virus, all of them contain the same ATCG nucleic acid component, right? So how do our immune system differentiate them? Because of the unique structure. For example, the methylated group. So for APT, for eukaryotic cell, normally we don't have methylated CH3 group. Probably in terms of the frequency of CPG sequences, which mean CGCG repetitive sequence, normally mammalian have very low, not many part of it. But for bacteria, they have a lot. So this is also could be considered as PAMs. So since there are a lot of different kind of PAMs, normally they are classified into different group. So these are an example of PAMs when it's involved other than microbial associated molecules, meaning the host tissues. So it could heat shock proteins. So these are the proteins our body secreted when there is unfavorable condition experienced by the